Hello and welcome to BioCode. In this video, we will see about Genbank data format or Genbank file format that is utilized in bioinformatics analysis. Genbank file formats are usually considered to be associated with NCBI because NCBI actually hosts the Genbank database. So any file that is retrieved in a Genbank format from NCBI or Genbank database is usually in Genbank file .genbank extension or .gb extension. So as you can see over here, I have Homo sapien lactase LCT mRNA extension, and the extension is NM0022299.4. And I click on complete record file and format Genbank full or Genbank. That depends upon your query or your need. So once the file has been retrieved in Genbank file, it could be .gb or .genbank. It really depends upon you. So let's visualize it in our code editor, or if you have Visual Studio Code or any other code editor, you can open it in Notepad as well, but that is not the proper way. Since at BioCode we recommend installing a code editor such as Atom or Visual Studio Code, that actually makes your life much easier if you are a bioinformatician or computational biologist. So right now I have a sequence.gb file, that is Genbank file, that is a file format basically to store information about the sequence or the molecule that we are actually analyzing. In FASTA format, the data is about either description. The first line is actually the description, the next line and the next further lines are the sequence, and that is that the file format is done. But in Genbank, there is a huge amount of information available about the sequence that we are actually trying to analyze. For example, we retrieved the mRNA sequence, but when we retrieve the Genbank file, it has a plethora of information, such as it has DNA sequence, the protein as well over here, and then more and more information. So let's go back to the top and let's start analyzing it. So the first question, why do we really need to retrieve a Genbank file, or what is that basically? Genbank file is usually retrieved when you need multiple information or when you need extra information about the sequence that you are trying to analyze. So in that case, for example, as